So on the previous class, on the previous class, we actually discussed. I mentioned, the part of the secondary messenger pathways and stuff. Once we start the next class on ester cycle and puberty, we'll go back to that. Okay, so even if we know at some point we have the other quiz, I will move that section out because I think it may just fit better there on the understanding of once we go into the whole control system, endocrine system, or the endocrine control of the ester cycle, we're going to get to that and might be easier. Meanwhile, just make sure that you understand that it's going to be covered at some point. So this is basically a half a class deal only on major pathways, hormonal pathways, all the way to transcription. This portion where we started to talk about, the follicular waves and, what is the folliculogenesis and the oogenesis, I think we mentioned before, one is the growth of the follicle itself. The other one is the maturation of the oocyte. Maturation of the oocyte, for all these reasons that I mentioned before, going to be the lone cell that will give rise to the embryo, and especially because at the beginning, is going to have to accumulate a lot of stuff in there. Okay, so from a cytoplasmic maturation point of view, this is what it is: accumulation of stuff. If you wanted to summarize, very easily, you have to accumulate nutrients, you have to accumulate mRNA, you have to accumulate buffers, you have to accumulate stuff. It's a ginormous cell. From a nuclei point of view, the maturation means meiosis. Parts of meiosis, I think that in summary, meiosis, progression. You know that, once females of many species are born, you're born with a set number of oocytes, and at that specific time when the females are born, you have all of those cells, all those primary oocytes, which is the oocyte surrounded by a few follicular cells. That oocyte is in a meiotic arrest. Basically meaning those cells, the oocyte started the meiosis process, but stopped. And will only continue after a few stimuli. And the stimuli will go on from being FSH and LH during the development of the follicle, all the way up to fertilization. Okay? So the sequence of meiosis will only occur once that, remember that little picture I draw in there of the follicles developing, that's where it goes. So accumulation of stuff in the cytoplasm, progression of meiosis on the nuclei. Very good? Excellent. Again, two things will not happen in the sperm cell. Okay? Simple there. Meiosis, bang, that's it. That's how much simpler it is for the male side. So this is already. So then we start the progression of the folliculogenesis. The difference right now in between this and the male, as I mentioned before, is that you're going to have this process of ovulation that only occurs after 14 days, 21 days, or 28 days, depending on the species. So there is this progression of things happening once, and at some point, depending on the species, you're going to have one, two, or several follicles ovulating. Okay? This portion here now means once the follicle starts to grow, the previous class, we mentioned that, and in the ester cycle class we're going to go over again, it is hormonally dependent, okay, all the way up to formation of that primary, secondary oocyte, or primary, secondary follicle. It's it develops. During the fetal stage, it's not dependent on any hormone or anything. After that, you start to, or these cells starts to be dependent on FSH and LH. Okay, so we mentioned before that they started as a group of follicles at the beginning of each estrogen cycle or menstrual cycle. And then from this big group, and let's assume right now that it's a species that only ovulates one. It doesn't matter if he ovulates more. The only thing is that from a bunch, just one or a few will actually ovulate, which basically means there is a certain selection of these follicles, from that big bunch. The big selection then, I think, and at this point, the only thing that's going to matter is that during that initial stage of this large cohort of follicles growing, it is FSH dependent. After is selected, this selection, we're going to go further in more detail, it becomes then LH dependent, okay? All the way up until ovulation. Only the ones that are selected becomes this antral, what we call this antral follicles, big antral follicles. There are some smaller ones, of course, they create that, that culi inside of the follicle, that space inside of the follicle, but they don't go anywhere. The other ones starts growing, and the difference here between this half of this picture and this half of the picture is that this is a growing follicle, almost what we call antral follicle or Graafian follicle. By the time it comes to this, and what is the difference that you see here? Is that these cells here in the middle are in the internal layer here becomes a little loose. Okay, this is one of the first characteristics of a follicle that is about to ovulate. Okay, and we're going to go into a few slides only about ovulation there. Okay, we went to full bodies, follicular structure. I think one thing that eventually, again, we're going to go back here. In this picture here, which is this little part here, just, augmented. We have what we call here the corona radiata, which is these very specialized cells. You could say a specialized cell or a subpopulation of granulosa cells or the cumulus offer cells. They're just two attached to the embryo, to the oocyte. And the oocyte I mentioned before is this whole, it's not this whole structure, but it's this internal structure. Okay, this is the actual oocyte. Cytoplasmic maturation occurring here, nuclear maturation occurring here. And this other layer here, which is comes out of nowhere, it's this glycoprotein layer that we call zona pellucida. The only difference there, or the major reason why this layer is here, is because it is extremely important for fertilization. Okay, we're going to see in the fertilization class, we're going to have a class only, or a set of slides only on that. It is just, important for fertilization. Very nice. Lots of more names for you too. This is the part where, before we go to the ovulation side, we mentioned that, these are the cells. In the case of the male, where I said that, you have the seminiferous tubule, you have those interstitial cells in between the tubules that we call Leydig cells, and those are the cells that produce testosterone. And the major thing is basically that. So they just go there, they receive cholesterol, androstenedione, they have enough or the right enzymes to turn that substrate into testosterone. In the case of the female, you're going to have all of this. So imagine that this follicle here is inside of the ovary, and there's this whole lot of, blood vessels around, is highly, irrigated. So you have lots of substrates coming in, either testosterone, androstenedione, or cholesterol. And what these cells have to do is to basically receive those things, or receive those nutrients, and receive those substrates and convert. And what happens is that it goes from testosterone and being converted all the way, or androstenedione to testosterone to estradiol. Okay? And in this case here, in this middle here, you might be asking, what's up with this middle? This middle is obviously very important because it is this environment where the oocyte grows. It reflects a lot what's in the blood because you have plasma coming inside. And this is where, people have done quite a bit of research in this area, how nutrition, how stress, and how other factors can affect the quality of these oocytes, and therefore the quality of this embryo that will be generated is affected in many ways through this. So you are in contact. If you have a lot of bad stuff going on in your body, it will show up here. Okay, so that's the whole thing. Obviously, all this estradiol that is produced by this follicle, you have a very, very high concentration of it here, much higher than in circulation. But obviously, you have blood around. Part of this estradiol is picked up by the blood vessels, and then it goes to the circulation. Okay? So steroid synthesis, this is probably okay for both male and female, but for us right now is like this. The estradiol, which is by far the most important one for that will be produced by the follicle, they receive testosterone from the theca interna, okay? And the theca interna has receptors for LH, okay? And this is what makes, that testosterone then being produced. This is the same LH that will then also control Leydig cells. So LH and testosterone are very tied together, okay? In this case. On the other hand, here you do have estradiol that will then receive. So this testosterone that is produced, if you go back here, testosterone is produced here, have a set of enzymes. It diffuses to this membrane, this basement membrane, goes to the granulosa cells, and through the action of this aromatase enzyme, it converts testosterone into estradiol, 17 beta. Do I need to write 17 beta? No. Fine. You're going to see during the technology classes there's lots of types of estradiol. I think we mentioned this in the molecules of each hormone, but for the purpose of this class, that's fine. So this is the steroid synthesis side of the follicle. Okay?
Does the theca internal function like the vertical?
It does in the sense that it's dependent on LH and synthesizes testosterone. So yes, you can say that it has lots of similarities there. Yes. Knowing about this, I think it becomes then a lot easier to understand once you have certain abnormalities, either in the female or the male, you can really shift testosterone and estradiol production quite easily. So you can have females producing quite a bit of testosterone, a lower amount of estradiol depending on the disease. Not the opposite goes for the male. You'd have to have like cells producing estradiol at some point in the testes, but that's also possible. Okay. Very good. So this general anatomy, general control of the follicle, what produces what as well, maturation of the oocyte, more or less. Okay, does that make sense so far? Yeah. We'll get this thing going, and we can go back and forth as we wish. After you have all of this, and I said, that half of that follicle picture is starts to go a little before ovulation, and I mentioned that, things start to become a little loose, especially in that granulosa layer. I think on the ovulation, the best way to perhaps, imagine what are the physical things that are necessary for ovulation is that, remember when I said before, for ovulation it's almost like an explosion. And in order for that explosion to be facilitated, obviously, it would be good to have some degree of pressure, something pressuring the walls of that follicle, which does happen. But the other one is that if you could weaken a portion of the wall of that follicle, that would be also desirable, because then you would have a place where that thing can get out, from the internal side of the follicle, and which is basically this stigma. We call it stigma formation. Is this the wall of the follicle becomes thinner, everything in the follicle becomes looser, and with that it becomes thinner, weaker, and at some point that's going to be actually the site of rupture that occurs during ovulation. So this loosening of this cumulus oophorus complex, prostaglandin role. When I say prostaglandin role there, the only thing is you have to remember in that case is that you do have a little bit of nothing to do or very little to do with the prostaglandin that is produced in the uterus. I told you before, prostaglandin is produced everywhere. But in this case, I think perhaps the thing to add here, if you can, is that it will act into these myoid cells that are around the follicle, or these little muscle cells. And the idea is that you can squeeze a little bit. So weaken the wall, squeeze. That's the two most important things that will have to happen, for the physical process of ovulation to occur. Okay? And obviously, it will be picked up by the ovulate. That's a given. I think I mentioned this, follicular fluid. And you might imagine, so Ronaldo, what happened to the follicular fluid once ovulation occurred? You have this massive amount of fluid, and it is a lot of fluid. What do you think happened to it? To leave. Okay. So what do you think happened? It goes to the oviduct. Imagine it explodes. Obviously, the oocyte with the cumulus oophorus goes to the oviduct. Do you think some of that goes along with that? That fluid? It does. Okay, so it's somewhat important that fluid also goes into the oviduct. It helps in that environment inside of the oviduct for fertilization. As I said before, it does have a bunch of things in that follicular fluid. Some of those are very, very intuitive. It looks or somewhat resembles what you do have in plasma, your blood. Obviously at different concentrations, but proportionally speaking, it does resembles a little bit of what you have in plasma. At the same time that you have the products of that follicular or those granulosa cells and theca cells inside. And surprisingly enough, obviously the concentration, unsurprisingly enough, the concentration inside of this follicular fluid is very, very high. Okay, of estradiol, for example, super, super high. And you do have a little bit of progesterone near ovulation. Even before, you do have quite a bit of progesterone. And then you say, why? Well, because during this process, you do have spillage out. You do have, like in the process of synthesizing, estradiol, you do have also a little bit of enzymatic activity, for example, to produce a little bit of progesterone and a little bit of some other things that are something that comes from the theca interna not all that substrate, not all that androstenedione or testosterone is actually converted into estradiol. So you do have a little bit of testosterone there as well. By far, to us, this is what matters. We say, yes, there's a lot of estradiol there. That's what is going to then, the estradiol, and this is the first thing for your ester cycle class, the estradiol will then be the major trigger of ovulation. Estradiol will have to go up quite a bit to a point where you have, the synthesis or the surge of LH and then ovulation. Okay, so another thing, the other part that, in terms of this follicular fluid, and this is not only the function of the follicular fluid, what is written there, but this is the function or what happens to those cells after ovulation occurs. So you do have this very big valve, and all of a sudden it explodes, so it shrinks. And whatever is left in there, which is a little bit of follicular fluid, all of a sudden, because of this explosion, this drastic explosion there, the oocyte, you do have a little bit of blood mixing in there. And that soup that was formed, in that inside of that leftover of the follicle that just ovulated is what is going to give rise to then the corpus luteum. Okay. This is, in a nutshell, of this folliculogenesis side. This is what is the basis of your estrus cycle, menstrual cycle as well. So we're going to go into a lot more detail, especially because of the hormonal control. But this is a nice summary of those. So at least you know more or less how the follicle grows, what happens to the oocyte, all the way to ovulation, and then after ovulation, preparation for corpus luteum formation. So then that's exactly the point where you shift from estradiol production to progesterone production. Okay? Very nice. So this is the basically a picture that describes everything that we went through. Primary follicle develops. At this point, as opposed to the first class, you should be able to start looking at this picture, and names have to pop in your head. Thick interna, thick externa, basement membrane, granulosa cells, oocyte, zona pellucida, nucleus, maturation, progesterone, estradiol, Okay? So you have to start putting those things, those detailed things together. This is the, when the very dramatic scene of the ovulation occurring, and then formation of the corpus luteum, and then eventually it should, the species or the female does not become pregnant, then this corpus luteum then becomes atretic and the cycle continues. Again, imagine all of this happening in one specific spot. Okay? Things do not travel around, like that in the cortex. It's just a specific spot in the ovary. And does it occur then here, or here, or here? Yes. Wherever those primary, follicles are, it's where the thing will develop. So the whole cortex eventually will be popped, by ovulation. No, that's okay. So this is the ovulation. This we already mentioned this for this pre-ovulatory follicle to go up until ovulation. We mentioned cytoplasmic and nuclear maturation, eruption of these cells, so they become more loose for stigma formation, and then actually the actual ovulation there. This is just a more or less of a curiosity. Most of the domestic animals, their ovary is like this, as I mentioned, cortex, medulla. The mare is the opposite. They have the actual, all the supportive structures in the cortex, and the actual follicles developing in the medulla, which basically means the shape of their active ovary is a little weird because they can only ovulate from one specific place where the oviduct is there. They call it ovulation fossa, but again, this is a detail, okay? So this is just a curiosity at this point. I'm going to use this as the basis for most of the questions that I have. Very good. This portion here, that we need GnRH and LH and progesterone and prostaglandin. The only thing that I mentioned in terms of hormones at this point is that prostaglandin will be needed for some of the contraction, for the ovulation process, and that estradiol is the major trigger. For this part of the class, that's enough. Once we go to the astrocycle class, we're going to go into all these things on LH and FSH and what you need for hypothalamic control of ovulation. Is that okay? But good to have a good read there. Very nice. So before we go into spermatogenesis, that you're gonna laugh at me, how much easier it is, the other part. But just keep in mind, we're gonna go back to most of these things in the astrocycle class, but from a purely follicle development, oocyte changes, and all the way up to ovulation. Does this make sense? Okay, a weird process, very different than the male, but it has to at least make this bigger sense. And again, another set or package of names that you have to add, to the class. Is that okay? Yeah, very good. So let's go then to the spermatogenesis. And the spermatogenesis, so this is, I'll go back to the previous slide, but this is let's go back to the testis, and then all the ductile or the duct system, of the male reproductive tract. And we mentioned that, the primary unit of the testis will be the seminiferous tubule. So the seminiferous tubule then is followed by, all of this. The seminiferous tubules are all around here. So basically meaning if you have a section of it, you're going to have all these tubules, one next to the other. And eventually, the lumen of them will be filled with not mature, but at least anatomically speaking, sperm cells, fully formed sperm cells. And in between those cells, you have what we call interstitial space. Okay? This is really the major division we make of the microscopic phase of the testis. Seminiferous tubule, interstitial space. Seminiferous tubule, where spermatogenesis will occur. Interstitial space, where steroidogenesis, in this case, testosterone synthesis will occur. Everything that needs to be supportive structures for the testes will be obviously in this interstitial space. Nerves, vessels, blood, nutrients, oxygen, all of this. And whatever that comes from the body will then be diffused to the inside of the seminiferous tubule. Okay? And then from then, the rest of the place, and for practical purposes, the epididymis, the body, the head, the tail, the vas deferens, the pediform plexus, it doesn't matter right now to us. We're going to focus on those guys. Okay? Now, a few things in terms of cells. At the same time that you have the primary follicle, secondary follicle, you have the obviously then the antral or graphene follicle, the oocytes, and those are the cells that we talk about, in the ovary. Some of the primary follicles and stuff you can call follicular cells, but we talk a lot about a structure of the follicle, not necessarily a cell, individual cell. Here is going to be a little different. because all of these cells, especially, we mentioned in this slide two most important cells, the Sertoli cell and Leydig cell. Leydig cell is this one here that then produces testosterone. Okay, major responsible for testosterone production. As you mentioned, is going to be majorly influenced by luteinizing hormone. Okay, so somewhat of a similarity with the theca internal cells from the follicle. Okay, inside seminiferous tubule, you're going to have then the Sertoli cells, the Leydig cells, the Sertoli cells. The Sertoli cells will then be influenced mostly by FSH, by follicle again. Cell that is inside the testes and is influenced by follicle stimulating hormone. Yeah, it doesn't make sense. It was when people found out first on the female side, and then they thought, oh, the same. But now they help to produce sperm cells. And it's too late to change the name, and they said, what the hell? Let's leave it like that. Okay, so the idea then is that, that cell, as opposed to, I think you could somehow correlate those Sertoli cells with those cumulus offer cells or that corona radiata cells of the oocyte, the follicle and oocyte. Why? Because it is the cell that will give support for the synthesis of sperm cells, or the transformation from primary, and then we call there some spermatogonia or primary cell, all the way up to a fully formed spermatozoa. In order for that change to occur, we're not going to get into the details, but all the way from the meiotic process is very energy consuming, is very complicated, it's necessary for very specific, a very specific environment, cell environment. And the Sertoli cells will do that for those sperm cells. They will nurture the cell in a certain way in which they can go through meiosis. They can go from a 1 cell to 4 sperm cells, and that's how it is. So it is a very fascinating cell. The Sertoli cell does a lot of things, but for us at this point, is influenced mostly by FSH, and it helps in the synthesis of sperm cells inside the seminiferous tube. Okay. Now, these other cells here, we call it primary cell or spermatogonium, and this is what we, you're going to see more pictures, but these are cells that are around here in the periphery of the seminiferous tubule, and this is what we call the spermatogonium. Spermatogonium is what we call the germ cell. And when I say germ cell, is because it is a fully diploid cell. And this is the one that goes first through mitosis. So they're going to be always there, basically meaning there's always going to be replacements for those cells. But then the daughter of this cell will then become or go on to meiosis. Okay, so this is what it means. Like lots of tissues in your body, once they become like very specialized, you have germ cells because it is responsible for the continuation or making those specialized cells to be there forever. Okay, basically, this is the kind of stuff that would be somewhat, relatable to your primary oocyte or primary follicle. Your primary is not the primary follicle. Your oocyte would be the one, but let's say your oocyte could always divide, even after you were born. Because in the case of the female, that's what happens. Imagine that this population of spermatogonium Imagine that this population of spermatogonium was population of sperm was limited. Means at some point they're going to go through meiosis, but eventually they're going to end. So the female do not have that capacity to keep replicating. So the production of, which means the production of sperm cells, long as the testis is healthy, it will produce it up until the end of the life of that being. Okay. After that, obviously, part of the class today is to talk about the transformation of this cell, and that's what I meant by by this regular shaped cell or this regular cell, all the way to a haploid cell that looks like an alien. Okay, so this is the process of spermatogenesis. No follicle, no changes in drastic changes in hormonal concentrations, no ovulation. There's no like a process where you have like a drastic thing. Continuous production, and basically no cycle. There is a cycle, obviously, because most of our hormones, they go up and down a little bit even during the day. So you do have a cycle, but it's not like the one that you see in the female. Good. So talking about more, we'll go into this, but this is just more or less the talking about the amount of, I think here, the sperm cell concentration. This is the one that, depending on the species, again, you don't have to know this, but from each ejaculate, you're going to have in the billions of cells. Okay, see how crazy it is compared to the female side, where you're going to have one every so many days ovulating. In the case of the male, you're going to have billions of cells produced basically every day. Okay, so it's a startling difference. Again, when we go to the technology side, and I think this is the other side of this class, when we go to the technology side of it and what people can do, especially with domestic animals in terms of improving efficiency or genetics and so on, reproduction is one of those systems. There is this vast space for improvement. And that's why you can do embryo transfer, superovulation, artificial insemination. You can freeze semen to be used. You can collect semen from a bull here in Canada and export, Russia. And there it can be used then to fertilize an animal over there. Or you can basically create in vitro embryos or in vivo, and then actually also either put in a surrogate mother or commercialize and freeze and send somewhere else. It's a huge economy in the livestock industry around those things. So we're going to go through this later. This is more or less like the picture, a microscopic picture of the seminiferous tube. So lots of these cells here, very dark. These are what we call the spermatogonium. Some of these bigger cells here, what we call the Sertoli cells. And you can see, like in this cut, it doesn't mean that this thing is really all the way up, but sometimes you have a few features of when you do this light. But this is already some of the sperm cells. Okay, so they go really from this kind of cell to this kind of cell. And I believe the other thing that, from a spatial perspective, this is a much, much, much smaller cell than the oocyte. Okay, when I say much smaller, like imagine this would be an oocyte, and a little dot here would be a sperm cell. Okay, that's the amount in terms of change in size or in volume compared to the other one. So yes, you can produce a lot more cells, but it's because you produce smaller cells also with very limited cytoplasm and so on. If you want to draw or want to be more, okay, this, for example, here, this would be what we call, the interstitial space. Okay, this is clearly a vessel. This is what we call a Leydig cell. Okay? So this is where things are. Is very basic anatomy of the seminiferous tubule. This is more like it's an older picture, but it is a bit more educational than the previous one and even this one here. The idea is that what's showing here is that this cell here, which we call the Sertoli cell, it's a very you can find in different places, different pictures. It's a very weirdly shaped cell, and it's very kind of amorphous. It will change as spermatogenesis go through it. You can have those germ cells that will go on all the way to the next stage, to the fully grown, sperm cells. So basically, your spermatogonium, then your primary spermatocyte. You can have your secondary spermatocyte. At some point, you have something that we call spermatid, and the next one, spermatozoa. That's the sequence of events in terms of changing of the cell. You can see here that compared to what we saw, or compared to what we did, how we look at on the female side, we also have somewhat of a nuclear maturation and a cytoplasmic maturation. The difference here is that the nuclear maturation of the female was about progression of meiosis, but in the sense that you have arrests in certain stages. So you start, then you stop, then you have another stimulus, you continue, and then you stop, all the way to fertilization, which is the very, very end of meiosis in the case of the oocyte. In the case of the male, or the sperm cell progression, what you basically have in there is the normal progression of meiosis. There's no arrest during the process. You just have, from a spermatogonium, as I said, is the germ cell. You have mitosis. This one stays. This one moves to meiosis. Once this one here is, determined to be or to start meiosis, that's it. It will go from that one cell. That's the other thing that I mentioned before. From that one cell, you're going to form 4 sperm cells. Okay? Why? Yes, it's efficient. And second, there's no need for cytoplasmic or the cytoplasmic accumulation. Do you have, or can you say that you have somewhat of a cytoplasmic maturation? I think you can, because you're going to have to highly transform that cell. So you're going to have a cytoplasmic modification, but it's not of the same nature of the cytoplasmic maturation that occurs inside of the oocyte. The oocyte is about accumulation. Sperm cell is about transformation, and actually, you want to get rid of cytoplasm. Okay? Yes? So that's the thing. The spermatogonium is going to be the only one there that will do actually mitosis first. Then one stays, the other goes. Yeah. So one stays, the other goes. Okay. So as you can imagine, it is a tissue where cell division goes like bananas. It goes crazy. Okay. A lot of metabolism, lots of energy being consumed, okay? And it's obviously the testes and the ovaries, they are highly irrigated. They consume a lot of, they have access to a lot of blood, which means oxygen, nutrients, buffers, and so on. Okay, here, spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, spermatozoa. Don't worry about, the only thing that I want you to pay attention here is that It's a diploid cell, of course, is the germ cell. Diploid. This is already starting meiosis, but there's still diploid cell. And then only spermatid and spermatozoids are actually haploid cells. Don't worry about this AB, APB here, but it has to do with a little bit of what you asked. Some of these types, somehow they have a cellular signaling there, which indicates one will stay, the other will go. So they are predefined where they go, okay? I haven't shut up, right? Nice. Okay, we have 5 minutes. Again, going back, spermatogonium, mitosis, spermatogonia, and then all the way from there, primary, secondary, spermatid, spermatozoa. It is basically a very good sort of illustration of what happens, and they don't hold back on cell division as the female side or the oocyte does. There's no accumulation in the cytoplasm. There's no formation of polar bodies. There's nothing of that. This is simply a meiosis process. Yes?
So is the secondary spermatocyte haploid or diploid? This slide shows haploid.
Oh, I see. I think it depends on. You can say haploid. I'm sorry. You can say haploid. So you can basically pick like the. I think that's where the other one on they say haploid, but you have like two copies there. I think it depends on how you, whatever you look. I'm not gonna be being too picky on that, but this is basically the secondary spermatocyte is when you start to basically going and dividing, still having the last division. So, but it's from the same copy. So that's why it is a diploid cell, but it has the same copy of the same strand of DNA. So that's it's technical detail, don't worry. So this is again another one to go and look at the table, just to take a look on this. Again, this is the part where why don't we show this a lot in terms of the female. The female goes through this, but stops, and is a lot easier. Now, this is the progression of cells. So up to this point, it's really like as long as you know well the seminiferous tubules, the cells, the spermatogonium, that you're going to give rise to the spermatozoa. You have 2 or 3 cells in the middle. Meiosis will go through. No hold back on the meiosis process. No cytoplasmic maturation. There's no cytoplasmic maturation, but there's transformation. And obviously, there's no such a thing as ovulation, anything that resembles ovulation. Every now and then I try to compare the two systems, but ovulation, there's nothing there. The spermatozoa, as I mentioned before, it will have to go from this cell here to something like, like this. Okay. So you will have to go from a very rounded cell, regular looking dude, okay, to that thing. And which basically means at this point, I think the best way to look at, obviously, these are names, another package of names for you. Congratulations. So the only thing here is that what you have to imagine is that from a cell point of view, you have to imagine, or going back to your the drawers inside of your head and say, so what's inside the cell? And what has to be transformed in this cell? You're going to have the nucleus, and then you're going to have organelles around. The cytoplasm is made of mitochondria. You have the endoplasmic reticulum. You have the Golgi apparatus. What else you have inside the cell? Vesicles of all kinds that are probably transporting things around, being fats or proteins. What else? What makes the cell have a certain shape? What holds the cell with a certain shape? Cytoskeleton. Okay, so there's all this kind of like fibers, micro. You could always say like little fiber that keeps the cell with a certain shape. So these are the things that will have to be transformed in that cell. If you look at the nucleus, for example, you basically say, is it going to contain your haploid nucleus at that point? So this is the stuff that will have to be actually the whole reason for the existence of the sperm cell is that it has to transport that nucleus to the egg. That's the sole purpose of the sperm cell. Okay? What is his mission? Yeah, transport the nucleus. That's the whole thing. The rest of the stuff, you're going to see that, that guy, if you look at the shape of that guy, and we'll continue in the next class. But that guy, if you look at the shape of that guy is made to run, swim, okay? But it's made to swim, which basically means you have to go through stuff. And that's the reason why, some of the other things that are in that cell is to actually destroy barriers to make sure that you can deposit that nucleus inside of the egg. Okay? So we'll continue from there next Tuesday. Have a great weekend. Don't drink too much. And